My name is Joshua Mugabi. I welcome you to topic 7, Inheritance and Evolution. We are on learning outcome A. Analyze the structure and functional significance of nucleic acid in meiosis and mitosis, their role in cellular functions, and how mutations in nucleotide sequences can contribute to disease like cancer. The learning outcome, we are looking at learning outcome A. Group of learners search for and discuss the 3D model of deoxyribonucleic acid, DNA, ribonucleic acid, RNA, and chromosome and base pairing rules. Create 3D model and present their finding in a gallery work. This time we're going to look at the 3 model of DNA. We are exploring the double helix. Item 1. Your class receives a 3D printed model of DNA. The teacher asks you to examine its shape, color-coded bases, sugar phosphate backbone, and twisted ladder structure. Task: identify the structure components of the 3D model. Explain why DNA is arranged in a double helix. Describe base pairing rules using the 3D model. Explain how the 3D model helps in understanding replication. Let us start with identify the structure component of a 3D DNA model. The structure components includes the sugar phosphate backbone, that is a side rail. The nitrogenous bases, we have adenine, thymine, cytosine, and guanine, and a double-stranded helix. Task 2. Explain why DNA is arranged in a double helix. Reason for the double helix. The 2 strands twist for stability. Hydrogen bonds hold bases together. Protects genetic information from damage. Three. Describe base pairing rules using the 3D model. Base pairing rules: adenine pairs with thymine, and these ones, thymine and adenine, are bonded by 2 hydrogen bonds. Cytosine pairs with guanine. Cytosine and guanine bond is made out of 3 hydrogen bonds. Four. Explain how the 3D model helps in understanding replication. Understanding replication. The model shows complementary strands that leads to easy to demonstrate strand separation. You can visualize how each strand serves as a template. Helps explain semi-conservative replication. Now let us look at the 3D model of RNA. And here we are going to investigate RNA structure. This is our item 2. Students analyze a 3D model of RNA displayed on a tablet showing a single strand, ribosome sugar, phosphate groups, and uracil bases. Tasks: identify differences between 3D DNA and RNA models. 2. Explain why RNA is single-stranded. 3. Describe the base pairing rules in RNA. 4. State how RNA's 3D structure supports its function. Let's start with identify differences between the 3D DNA and RNA. Differences. RNA is single-stranded. DNA is double-stranded. RNA contains ribose sugar. DNA contains deoxyribose sugar. RNA has uracil instead of thiamine. RNA structure is shorter and more flexible. Number 2, explain why RNA is single-stranded. Why single-stranded? Needed for diverse functions. Messenger RNA for coding, transfer RNA for transport, ribosome RNA for structure. And this also allows folding into various shapes. For example, clover leaf and loops. Three, describe the base pairing rules in RNA. Base pairing rules in RNA. Adenine pairs with uracil. Cytosine pairs with guanine. Four, state how RNA's 3D structure supports its function. "3D structure and the function." Flexibility allows RNA to fold into enzyme-like shape, ribozymes. Single strand enables temporary roles in protein synthesis. Transfer RNA's 3D shape fits amino acids. Now let us look at 3D model of a chromosome. And now here we are looking at chromosome during mitosis. This is our item 3. In the biology lab, a giant 3D chromosome model is shown. 2 chromatids, centromere, and a coiled chromatin fiber. Task: identify the main parts of the chromosome model. 2. Explain how DNA is packaged to form a chromosome. 3. Describe how the chromosome model changes during cell division. 4. Relate the structure to its function. Let's start with identify the main parts of the chromosome model. Main parts. It has 2 sister chromatids. It has a centromere. It has heteromeres and supercoiled DNA wrapped around histone proteins. 2. Explain how DNA is packaged to form a chromosome. Packaging of DNA. DNA wraps around histones. This forms nucleosomes, which coils and then supercoils, forming chromatins, which form chromosome. Number 3, describe how the chromosome model changes during cell division. Changes during division. Chromatin condenses during prophase. Sister chromatids separate during anaphase. Chromosomes decompose in telophase. 4. Relate the structure to its function. Structure-function relationship. Tight packing protects DNA, allows accurate separation of genetic material, prevents tangling of long DNA molecules. Let's now look at base pairing rules. We are going to be completing the base pairing. This is our item 4. Students receive a card set showing 1 DNA strand. Adenine, thymine, thymine, cytosine, guanine, adenine, cytosine, thymine, guanine. Task 1. Write the complementary DNA strand. 2. Write the messenger RNA strand transcribed from the DNA template. 3. Explain why base pairing rules are essential. 4. Predict what happens if base pairing errors occur. Let's start with write the complementary DNA strand. complementary DNA strand. Thymine, adenine, adenine, guanine, cytosine, thymine, guanine, adenine, cytosine. Rate the messenger RNA strand transcribed from the DNA template. Messenger RNA strand, uracil replaces thymine. Uracil, adenine, adenine, guanine, cytosine, uracil, guanine, adenine, cytosine. Three, explain why base pairing rules are essential. Importance of base pairing: ensures accurate DNA replication, guarantees correct transcription leading to correct proteins, conserves genetic information through generations. Four, predict what happens if base pairing errors occur. Consequences of errors, mutations. There will be wrong amino acids in proteins. There will be genetic disorders, cancer, or non-functional proteins are likely to occur. Now let us look at the transformation of DNA to RNA to protein with the 3D models. This is our item 5. You rotate through 3 stations. Station A, 3D DNA model. Station B, 3D RNA model. Station C, 3D chromosome model. Task: describe how information flows from DNA to RNA using the models. 2. Explain how improper base pairing disrupts this flow. 3. Use the models to demonstrate mutation effects. Describe how information flows from DNA to RNA using the models. Information flow. DNA in chromosome stores genes. When transcription takes place, they produce RNA, which translates in forms proteins. 3D models show structure steps from a double helix to a single RNA strand to a condensed chromosome. 2. Explain how improper base pairing disrupts this flow. Improper base pairing disrupts synthesis. Wrong codons will result to wrong amino acids, which will result in defective proteins. 3. Use the models to demonstrate mutation effects. Mutation effects show a swapped base in the DNA model. Demonstrate changed complementary base in RNA. Explain altered protein outcome. Thank you very much for participating in EVOLUTION and the evolution. We have been looking at learning outcome A, analyze the structure and the function significance of nucleic acid in meiosis and mitosis, their role in cellular function, and how mutation in nucleotide sequence can contribute to disease like cancer. We have been looking at learning activity A, whereby the learners search for and discuss 3D models: deoxyribonucleic acid, DNA, ribonucleic acid, RNA, and chromosome and base pairing rules. And we were supposed to create 3D models and present our finding in a gallery work. Please, if this information has been useful to you, visit me at Mugabi Joshua Biology on YouTube.